Abhijit, the link is ready. Who is taking care of Insta and YouTube? YouTube, you're taking care. Instagram? Chandana?
Sir, Lochna is asking me to do so. I'll just see once if I'm able to do it.
Yeah, only in case if Lochana is not able to manage. I believe she has a fluctuating network. That's the reason she wants a backup. Okay?
Yeah.
So better you be ready. If she drops out, then you can take it.
Yes, sir.
Okay, YouTube and Insta link. Can we share it, Soumya, to the participants?
YouTube links are sent, sir.
Okay, that means they can view now.
Yes.
Okay. Chandana, no problem. Instagram.
Yes, sir. Yes. No problem.
Okay. So YouTube live streaming, please ensure that there's no problem because last session we faced problem. But Abhijit, I was told the suggestion that you back the time, that also didn't work. Many were complaining about that. Okay. Okay. But what was the number participants who were on to? That is too less. Okay. Anyway, I think we will hope. But probably they were discouraged because of the fluctuation. I hope it should not happen this time.
So, should I record the session?
That would be nice. But how would you do?
I can record, but I don't know how to send because you can't send through WhatsApp. I can record on screen.
Exactly. And any video, let it be on our NSA YouTube channel for future use. And
fine. I can do is screen record.
That's what. Because that's the alternative left now. Okay.
Yeah.
Mukta, Merlin, and Sumishal, documentation, you're ready? Merlin, who's doing it? Okay, Chandana, hand in hand, you also click the photos like you did last time for the previous session. And please upload all those photographs onto our Insta channel, Instagram post.
Yes, sir.
Good evening, all.
Good evening, sir.
Good evening, Viola.
Good evening, all. Good evening, ma'am. Good evening, Jay, sir.
Jay is eating nippato.
Good evening. Actually, I put the mic on mute and I'm talking. Oh God.
Nippato is now promoted to Opatu.
No. Now I think we are visible. My friend said all our conversation was that.
Oh, wow. Okay. Go mute now.
Jay, sir?
Yes, Kavya. Now you are audible and clear.
Is it audible?
Yes.
Yeah, now it's very clear.
Yeah.
How did you rectify, Kavya?
It's from laptop to mobile, sir. I switched over from laptop to mobile. Simple one.
Oh, okay.
So Kavya, after the moderation session is over, Teresa will announce your name and you thank the speaker. After that, Putul will do the final vote of thanks, thanking everybody, the overall and the final one.
No, vote of thanks, I'll hand over to Putul. Right, sir.
Putul. Yeah.
Yeah. And now this session I have switched from mobile to laptop. First session I was on a mobile.
Okay.
So Putul and Kavya are trying to reach the other ends. I cannot handle in mobile. I don't know why.
Even I am finding it difficult with mobile.
Yes, ma'am.
It's better for me.
Visible and it's little difficult. Yes, agreed.
Actually, I have made one mobile stand also to place the
Oh my
mobile, but
God.
still the screen is very small, no?
Correct.
Any idea why did YouTube lag behind in the first session?
Abhijit,
Okay. No, you should have reduced it to 240 only, man. No, but when too many people and presentation starts, no, it may become slow. You keep a track of it. If at all it again lags behind, you reduce it to 240. Yeah.
Thomas sir, Shivkumar sir, Reverend Prem Mitra has joined.
Okay.
Instead of Avinash, Pastor will speak about Arusha initiatives. So he'll be the welcoming and then we'll proceed.
Why Avinash is busy, huh?
He's in some field work it's him suddenly, so he couldn't. That's it. He
Okay.
requested Pastor to come.
Pastor Premitra is there?
Yes, sir.
Yes, sir. Yes, sir.
Okay.
Hello, sir.
Hello, Pastor. How are you?
I'm fine, sir. How are you?
Morning, I specially mentioned your name.
Yes, sir.
Morning, did you join?
I couldn't join.
Morning.
Sir. No, I couldn't join.
Okay.
We had a funeral service, so I had to go there.
Okay.
So, okay, sir. Nice to be with you all.
Morning, I especially remembered and mentioned your name.
Thank you, sir. Thank you very much.
Recently, you were in the group. My niece was
Has
telling
also
us.
joined.
Nitin, good afternoon.
Hi, Nitin.
Nitin has started presenting only, all of a sudden.
I also see Dr. Chandish Balal, former director, NBIR. Welcome, madam. Sorry for the first session glitches.
No problem.
Nitin, can you
Thomas,
hear?
yeah.
Hi, Nitin.
I think he's muted. He will start slow.
Muted but sharing the screen.
Yeah.
Nitin, can you hear?
Hi, Dr. Abraham Varghese has also joined. Good afternoon, sir. Welcome, sir. Sir, your mic is mute. Your turn. Yeah.
Now I've
Yes, sir. Audible.
So how are you? How are you doing?
Fine, sir. Fine.
Good.
Nice to see you, Dr. Abraham Varghese. I am Thomas here.
Oh, hello. Where are you? Oh, Thomas. How are you, Professor?
I am slowly becoming tech savvy.
Oh God, it's too simple. Don't worry.
Last time I was frightened to wish you. So this time slowly I am.
No issue. Yeah. So, hello.
You are becoming tech savvy. It means a sign of danger, huh?
Okay.
Dr. Abraham Varghese, you were in the morning?
No, I couldn't be there in the morning. We had the National Bureau, we had some, and after that, we had, that went for almost 2 hours and I can't view continuously. Then, and Dr. Reddy's program was at 2 o'clock, So I think my colleague Dr. Chandish participated in it. So
Okay.
she was there. So I always take a break. So I told Jay Shankar I would attend Nitin's program. So
Okay.
that's
Nitin is our old student.
Okay. Yes. Your products are all very good. So keep it up.
I know.
Yeah.
Products are better than the factory. Dr. Abraham Varghese, products are better than the factory.
Okay. That's good.
Hello, ma'am. Nice to see you. Sandesh Balal, ma'am. Hello, good evening.
Hi. How are you? Good evening.
Yes, ma'am. Same here. Good. How about you?
How are you? Yeah, I'm good. Yeah. Good evening, Varghese.
Chandish, how are you?
I'm good, sir. Morning
So
also
whole day
we
you've
could
been
not.
webbing.
I couldn't get into that. We tried to access through YouTube because this was full by the time I completed that meeting and came in here. So YouTube link, there was a bit of a problem. Internet issue was there. But partially I could view and
I see.
there was a lot of buffering issue which I informed Jaishankar. So let's see now maybe. So I was very keen that here I should get into this Google link so that I managed to see all the slides. There it was not tallying. The slides and the lecture was not. But it was an excellent presentation. I hope it is there on YouTube after this also. I don't know. I should ask Jaishankar. Yeah. If it gets uploaded,
we are
I would
working
like
on that.
to. Yeah.
We are trying to because the provision to record was not there.
No. Madam, you can check on the Natural Science Association channel.
Yeah, we did, but when it was happening live, there were issues. It was not only for me; no one could watch properly.
Yeah. Live was a little bit problem.
Yeah.
The audio and
The
video
slides
was
were
not
stuck. Yeah, the slides were stuck. And there was buffering
audio was
issue.
going on.
And audio also was stuck in between. So there was no.
Nitin, welcome. Hope you can hear us all the way from America. Thanks for coming to be one of the resource person. You got to unmute and then speak, I believe. Trissa, if you're there, we'll start. We have 2 more minutes.
Yes, sir.
Okay, it's 5. We are trying to reach to a speaker from South Carolina University. Tressa will start, and I hope Nitin will be able to present.
I welcome everyone present here to the international webinar on moth diversity, ecology and conservation, organized by Natural Science Association, Department of Zoology, St. Joseph's College, Bangalore, in association with Arosha, India. Before we begin the webinar, there are some guidelines that we kindly request you to abide by. Please kindly turn off your mic and video during the webinar to avoid any background noise and avoid presenting your screen. Whoever does so will be removed from the meeting. Participants who are live streaming this webinar on YouTube and Instagram, please type in your name and name of your institute in the comment box. The same applies for any doubts or questions. Participants who are using Google Meet for the webinar are requested to use the chat box to type in your name, your institution, your doubts, and your questions. The questions will be answered during the question and answer session. Certificates will be mailed to you within a day or 2. Our moderator will convey these questions to the speaker, and feedback forms will be sent to your mail. We will now begin the talk on moth ecology and
Tressa,
evolution.
1 minute. We'll try. Nitin is trying to connect. Nitin, can you hear me? Nitin? I think he's trying to connect to us. He is muted. I'm checking with him. I think Nitin is not able to hear us as well. I think this will take some time to set.
Jay?
Yes, sir. Yes, sir.
Whoever has scheduled the meeting, just check. Can they unmute Nitin?
That is Soumya. Soumya, could you
Sir.
try?
No, you can't unmute him.
Okay. But I am getting communication from him that he's not able to hear us.
Neither we can hear him.
Yeah, true. Yeah, he has now left and he said he would rejoin.
Yeah, that will be better.
But okay, we'll try that option. Soumya, you got to let Nitin in. I think we have one more slot left and Nitin could be permitted. Soumya?
Yes, sir. Yes.
Sir, there's one comment there.
Yeah, yes, come.
Nitin, you're able to listen, hear us? Okay, Teresa.
Yes, sir.
Yeah, we'll go ahead. We'll start with Reverend Prem Mitra. I hope by then Nitin could set his thing. Okay?
Now, Jay.
Yes, sir.
Somebody can make a WhatsApp call to Nitin and ask him.
Yeah, I just spoke to him.
Yes, sir.
We both, me and Teresa, spoke to him now. He had this issue with the Mac being used. I believe he shifted, copied to another Google Chrome format. So he is now trying to reach to us. So we'll have Reverend Prem Mitra speak and then proceed. Yes, Teresa.
We will now begin the talk on moth ecology and evolution by Mr. Nitin R. I request Reverend Pastor Prem Mitra, Chairman of Arosha, India, to welcome the audience present and share his initiative.
Good evening and a warm welcome to each one of you for this international webinar. It's been a great to be part of this webinar, especially co-organizing the webinars at St. Joseph's College. Thank at this time the principal and all the staff for this excellent webinars which have been helpful for each one of us. Something about Arosha. Arosha International, or the international, started in Portugal 3 decades back. And from there, it has been around 20 countries. In India, 2003, it was established mainly in working in the Bannerghatta region. Our programs are more on conservation science, hands down conservation especially. Arosha India has different programs on mitigation measures of reducing threat of human and animal conflict. Been almost 10 decades on this work, especially on the main mammal elephant. We also have different other education programs. We collaborate with institutions. We help students who come to us. We have a research center. It's about 25 kilometers, Bannerghatta, a journey to the Bannerghatta National Park. Students, not only colleges around India, but also international students come there. We have also a community program where we believe very strongly conservation it's only not just a part in the forest or wildlife what we work, but also to be part of the community life, which plays a very key role, especially in the conflict mitigation programs. We also have a center, which I have already spoken about. You're welcome to be part of this programs, volunteer in our different programs in us. And also, you can also visit us. It's not very far away. If you're staying in Bangalore, it's just close by. It's about 26 kilometers from Bangalore. Arosha has been an organization which has been working with not only in the field of, we are also, at this time of COVID, we have also taken a key role in helping people around, especially our people around the national park. So this is some of our programs. If you're interested, please do contact us. You can mail us or be part of us by coming and staying with us. Thank you so much for being with us this evening. And as once more, I'd like to welcome each one of you and thank the
Pastor, if you could elaborate some more initiatives globally as well as in India. By then, I'm also seeking time for Nitin to get back.
Okay. Jaishankar puts me in always a tough spot because
I'm sorry.
Russia started in a very humble beginning. Our initiative started in with a small few volunteers. Our strength is our volunteer strength. We have a very small staffing, but the number of volunteers come to us. Another important thing that Arosha is that most of the science organizations do expect volunteers to have skills, especially the background of science. But Arosha has got one thing that we would also welcome people of other fields to come to be part of our programs. Volunteering is one of the key things that we have. We started a program called especially for people in Bangalore. We believe that where we work is an important place where it's a tip of Eastern Ghats. As you know, Bangalore has been expanding, and the amount of way that is moving, and especially towards the national park. It's a joke that one of the advertisers said that if you stand in your balcony, you can watch the elephant. Yes, very much true. If the elephant passes by, yes, there is places where elephant passes by. So this is a very important place. So we started a program especially for Bangaloreans, Bangalore for Banargata, trying to bring in understanding of what conservation is all about with the different talks, taking people, giving them exposure inside some parts of this so that they understand what the biodiversity is. It's an opportunity for not only to listen, but to act upon. We have quite a number of programs that was the previous year, and we are planning to start that once again, where we have a small seminar and people come there to be in the hands down, like maybe they help us in different things that are happening around there. Volunteering us in some of the things of cleaning up the forest, like removing plastic, because as you know, there are a lot of waste that are thrown that ultimately goes inside the forest. You might have thrown it outside, but slowly it moves inside. So picking up the dirt, bottles, and certain things so that they help us, and they also help us in some of the main thing on the barriers that we have, the barrier survey. There are different barriers in Banargata. As you know, some of the barriers need to be rectified. So with the help of the forest department, we take volunteers. And recently, there's an old rail barrier, which is quite an effective one. So we've done some surveys on it with the help of volunteers, so that you need to walk the whole stretch to find out where the barriers might have a defect, to rectify it, and to tell the forest department that these things need to be rectified, or when there is no barriers there. So we try to work on certain things. We also have an effective program that which is the recent thing that's happening around the camera. We place cameras and monitor movement. There's a lone tiger which is going around in the national park, which we are trying to monitor. Through this, we have come out with a small mammal, a guidebook for the Banargata National Park, which is a unique one, which many of them people would love to work in Western Ghats, and but very few would opt to work in the Eastern Ghats. And some of the would be amazed to see certain things that are there which is not been found earlier. So through this camera traps, we are able to find out some of the new things which we could not sight by our eyes, so that cameras are used. And this program is going on very effectively. Even initially, we started off but our main work is with elephants, so that is where we were using it, and later found out it's be very effective to find out different animals. The leopards, sometimes what happens is there are the boundary, out of the boundary, there are some places where the elephant comes in and goes out, and so trying to find out some places where it's helping people around there. Maybe it's a close compound, like in home for the old people, which we had a regular visitor, a leopard used to visit. So you can't sight it very easily, but through these cameras, we could find out that how frequent it comes in and how is it able to come inside, and trying to mitigate this problem. What is the cause of it? Trying to help the community around that. So this is one more of this usefulness with the camera traps that are happening. We're also partnering with other NGOs, trying to the whole planting of saplings, which has been an effective process, not only outside the boundaries, but also some other part of the boundaries, which is the buffer zones that we call it, planting saplings there with the help of volunteers, which has been a very effective program, which has got many volunteers to come in, and it's a tough thing when we realize that many of them would like to plant a sapling and then forget it. So our program says that you need to maintain it. So we have a program called to plant a sapling and then trying to maintain it. So if they are not able to come there, we would love to be part of it. So
sure.
Thank you so much for.
Thank you,
Nitin
Pastor.
Welcome.
Yeah, Nitin is here. I'll just try if we could hear. Nitin, can you hear me? Am I audible? He's able to share, but he's having problem with audio. Okay, I'll get his connection there. Participants, please bear with us here as well as those viewing the YouTube live streaming. These are technical glitches. Nitin is operating from there from the US. That will get set right within few minutes. I also would like to invite Dr. Abraham Varghese. Sir is a renowned entomologist, former director NBIR. Sir was also the head of the Division of Entomology in Indian Institute of Horticulture Research. Vast experience in both birding as well as in entomology. Sir, there are some thoughts that you would like to share on moths and insects to the participant for the benefit of the participants.
Okay. Anyway, thank you for the opportunity. I hope I'm audible.
Yes, sir.
I'm audible. Okay. I hope Nitin could resolve his issues early and kind of come in as early as possible. Maybe till then, let's see how best I can bridge the gap for a while, for a short while. Just giving you an intro on moths per se. Now, I think all of you know about moths. Moths are lepidopterans. They have wings, two pairs of wings, and they are called lepidopterans because they have scaly wings. You catch a moth, and the scales rub off on your finger. Now, when you think of moths, you should think also of the butterflies. Both of them are different. Moths are generally nocturnal, and you will, and they're dull colored, and their antennae. Insects have a pair of antennae which they are quite varied. But in the case of a butterfly, which is also a lepidopteran, the antennae is pointed with a distal end, club shaped. Now, most of you may be knowing this because you all would have studied basic entomology. Now, I've been an agricultural entomologist, and for me, from the perspective of moths, I see them on two fronts. One is, let me talk the positive side of moths. Okay, first, now all of you know, all of you love silk saris, silk kurtas, or sharwanis or whatever, and that is from a moth. So that is the beneficial side of a moth. So, for example, all of you may be familiar with sericulture. See, it's gone into a culture, it's gone into a subject. Degrees are offered or based on a moth. So you have BSc Agriculture College offering sericulture. Even some of the other universities, including Bangalore universities, used to offer a sericulture program, master's program. Lot of research on sericulture, and sericulture is the backbone of many people's livelihood. Now that is all about moths. It's because of moths. We have to be extremely thankful to moths for that. Now, another important dimension, and there are several wild moths also, which give us silk. The Irai silk moth is there, and quite a number of them in India. Now, in fact, we have a silk board based on this moth. So that's the power of this moth. Now, another important dimension about moths is that they are very good pollinators. They are very nocturnal, and the adults, the younger stages are always called the caterpillars. Then they pupate, and then the adults which come out are moths, and they are nocturnal, and it goes undocumented. There are several kinds of moths which come to flowers and affect cross-pollination almost from dusk to dawn. Now, many of the cropping plants and the flowering plants and the forest plants have their anthesis sometimes, say, between 4 a.m. and, say, 8 a.m. See, the early morning pollinators are all moths, mainly moths. It's only when the sun comes out that the bees also come out. But the bees get greater credit than the moths. So we need to look at moths. Nocturnal moth study is a very interesting dimension. Now, if you want to really watch these moths, you should go to a place where it is literally dark, not in the cities. Put on a light, and you'll find several moths coming. They are very phototropic, as we say. They come there, and that's the best place to collect the adult moths. You can collect them, and probably the earlier talk you all had in the afternoon perhaps would be the right place and the person to get your moths identified. But they are very diverse, very interesting to see, and they have, as I mentioned earlier, they have dull colored, but the patterns are very striking. And some of these moths have the hind wing very brightly colored, to ward off predators. So the third important beneficial aspect of these moths is they are an important link in the food chain. Now, if you take the food chain, there are many insectivorous, many insectivorous birds, many insectivorous mammals, and many nocturnal feeders. For example, we have the owlets, small owlets, smaller one, Athene Brahma owlets, they feed on big insects. And these big insects are needed to keep the birds alive, some of the night mammals alive. There are insectivorous bats also. So that's the important part. It is an important link in the ecological food web. Now, another fourth dimension of the moths is the larvae of the moths, the caterpillars. are eaten. They are edible. And in many tribal areas, even in the Northeast and across many countries. Just about 10 days ago, Dr. Jaishankar knows, I gave a talk on insects as food. And one of the important foods is insect food. For example, there's a worm called the bamboo worm, which is a moth again. And that is sold in Bangkok for a premium because that food is rich in protein, and these insects contribute to the athletic build and energy in athletes who are competing. So insects also as food, and especially moths, have a dominant part. Now these are some of the plus points of the moths. Now let me tell you the other part of it, because we need to know. Now we have several moths which are insect pests. We have Spodoptera litura, we have Helicoverpa armigera, we have Coursera cephalonica, Cytotroga cerealia, both on the stored grain and in the field. And once they come into the field, it's extremely difficult to control them. And the losses vary from 30 to 100%. That's how it is. Now, so they have been working more on the economic entomology part of it, and moths are, especially there is a family called Noctuidae, there is a family called Pyralidae, there's a family called Gellichidae. These are all important families which are pests. Then another important dimension is moths can fly like birds, butterflies too, and most of the insects can fly, but moths can fly a big huge distances. That's troubling us. Now, for example, recently all of you would have heard about the fall army worm, which is Spodoptera frugiperda, which attacks maize. Now, maize is an important crop in Africa, as well as in many parts of Asia. And we grow corns as food. Now, the most important thing about and we believe it is from the west side of India, maybe Africa, where we don't know, but it was first seen in South India. Now it has spread all over India and it has gone up to Nepal. And the loss is huge in crores of rupees. And the important thing about frugiperda is we cannot spray insecticide easily, because maize is grown as a fodder maize also. So on a fodder maize, if frugiperda Spodoptera attacks, this moth attacks, you can't spray because you're feeding it to the cattle. So we are stuck. We are stuck on one dimension of the pest management. So a lot of entomopathogenic nematodes and entomopathogenic fungi and all these come into picture. Another important moth which came into India in 2014 is Tuta absoluta. It's called Tuta absoluta. It's a borer on tomato. Now, these are all specific pests. One is on maize. Now, this is on tomato. And, it makes pinholes on tomato, and the whole thing rots. So, we've been able to develop some pheromone traps, and we've been able to get into the management part of it. But I would like you all to be seized with this important issue that we face invasive moth infestations also in the country from an India perspective. And this, for example, this Tuta absoluta originated in, say, somewhere in South America. It moved into the European area, into the Mediterranean, and somewhere they say it moved into Balochistan, and then perhaps Pakistan, and then came into Gujarat, and then into Maharashtra, and all over, So that way, these moths are capable of good movement, and the wind helps them to also move. So moths, therefore, are economically important. Yeah. So, Jay Shankar, this is a good, I thought a small preamble to Nitin's talk. He will be talking more on the ecology and evolution, and so I won't be able. I think that's if anybody has any questions, you can ask her. Or without a preparation, I won't be able to talk too long. So these are the thoughts that came in.
Sure, sir. Thank you, sir. That itself is big
Any
information.
questions? Pardon?
Yes, sir. That itself is a good information. If audience have any question, you can ask Dr. Abraham Burgess about this. We are also looking at the other dimension of mods. Parallelly, what's happening is Nitin would be sharing his slides. He would be talking over his mobile, and we'll place it, try to make it audible to you, and also run the slides. This is going to be a bit of the slow movement, but please bear with us. The information will reach you.
So Jay,
2 minutes.
we can ask all the participants to mute themselves.
Okay.
Listening to the talk, ask all the participants to mute themselves.
Okay. All participants to mute so that there's no background noise, there's clarity once Nitin is speaking. 2 minutes, we'll start with it.
Okay, good, very good.
Hello, Abraham sir, am I audible?
Yeah, you are audible.
Yeah. What is about pheromones, sir? If some insect pests that are causing crop loss, if we can use such a type of pheromones in the crop field, then we can control the pests, isn't it?
Very well. In fact, in Tuta Absoluta.
Please throw some light on that pheromones. What are the pheromones and which type of pheromones that are produced by different type of insects, like that, please?
Yeah. Who's asking me the question? Who's asking me the question?
So this is Kiran Nagon.
Okay, Kiran. Okay.
Thank you.
Okay. I think you were there for my talk on edible insects also.
Yes, sir.
So, okay, and a very good question. See, so let me put it very briefly because Nitin may come anytime, no? So, pheromones are nothing but chemicals, number one. The second important point is insects have a very strong olfactory sense. That is the sense of smell that is very strong. Okay. Now, number three is it is pheromone which communicates or disrupts communication in insects. So and in this case, what I was referring to is a sex pheromone. The female releases that pheromone and the male comes to the female. And they mate and they lay, and the female lays eggs. Now, what is done is the female pheromone is identified, synthesized, and put in a trap. Okay, so I have developed a sticky trap for this pheromone, and the males come and get stuck in the sticky trap instead of going to the females. And the unmated female will just perish without laying eggs. So that's how the control takes place. Okay, so and a lot of literature is there. You just Google insect pheromone, you'll find a lot of it, and we have more than 24 pheromones actively commercially available in the country. Okay, so. And pheromone has been one of my main areas of work in fruit fly in which is another pest. Okay, in insects. Okay.
Yeah.
So, any other questions?
And I too want to have some doubts on coffee borer. Coffee stem borer is the common pest here in Chikmagalur. And if it is also able to controllable through the pheromone, then it's very, very nice here to control the pest.
Yeah. Yes, you're very correct. If it was that's the dream that's the dream of every coffee entomologist. But unfortunately, pheromones are available for coffee stem borer, and this pheromone traps are placed in the coffee estate. It's subsidized by the coffee board. It's placed in the coffee estates. And one good thing it does is because this coffee borer comes in waves. See, suddenly the population goes up, then comes down, goes up and comes. So when the population fluctuation is easily discernible in the coffee stem borer pheromone trap. It is available. It is available. So it is sold subsidized by the coffee board also. So when the coffee estate, there's a manager or whoever, he detects the coffee borer in the trap. That's the time he will time his spray on the coffee. So to that extent, surveillance and prediction of the population, the pheromone trap helps in case of coffee stem borer. Okay. Yeah.
Thank you, sir. Thank you so much.
Welcome. I must thank
you,
Professor Thomas Sagre also for giving me this opportunity.
Thank you, Dr. Abraham Varghese. It was a wonderful session. I too enjoyed.
Yeah.
I really
so
enjoyed.
Thank you. Thank you.
Abhijit, you're there? You can hear us? Has Nitin connected to you? Okay. Will he be audible to the audience? We'll move the slide also. Yeah. Is that Nitin? Okay, Nitin, I hope the participants can hear. We will, that's very feeble, but I hope it's able to reach you. Nitin, a bit louder. That's Abhijit. Can you facilitate that so that we can have the slides going and Nitin talking to you over the speaker there? Audible. Nitin, that's fantastic. Start. Yeah. Yeah, Abhijit.
Hello.
Abhijit, that was audible from Nitin. We will go with that. Yes, Nitin, you please start. Nitin, some more volume, like the way you started. Abhijit, you can convey that to Nitin. Abhijit, can you stop for a while? Abhijit, can you hear me? Can you ask Nitin to hold on? I'm getting messages from YouTube. People are not able to hear. Can something be done, or we ask them for alternative? YouTube not possible. Okay. Proceed. Let's. I think we are not able to hear Nitin. Okay. Shall we try that, Abhijit? Yeah. Dear participants, please bear with us. We will take a small break. This is to ensure that the video and audio go hand in hand. We are making an alternate arrangement. I request Abhijit and Soumya to coordinate so that whatever PPT Nitin is sending, you can play it from here and he only speaks. And also there is grievance from the YouTube viewers that they're not at all able to hear. Can something be done to them? Yes, thank you. I request the participants to stay. The feedback form will be sent here and the YouTube link. We are trying to work out an alternate meeting through Microsoft Team. We will send the link here. You can all join there. So please bear with us. It will take few minutes. We don't want to cancel the session nor disappoint you. This will go on as scheduled. Yes, Abhijit, tell me. Nitin, ask him to speak. Let's see. Yeah, audible. We'll have to check with the YouTube people. If not, Rachel is setting a Microsoft team meeting. Abhijit and Nitin, Microsoft Teams meeting is getting ready. We will send the link shortly. We will all shift there so that all these glitches can be avoided. Because is that okay? Okay. Okay, Nitin, because we don't want to disappoint you or the participants. We respect everybody's view here. So please bear with us. This talk will go on. Nitin has made an excellent preparation and the content is very good. We are very apt. So we want to hear to Nitin. So the changes, we will now post the link. Everybody join there. Please hold on till then. Dr. Sabita is here.
Hello?
Madam, you can introduce, tell about Nitin. We missed that part of introducing Nitin because of the connectivity issue.
Sure, sir.
Yes, madam.
I am very happy to introduce Nitin Ravikant Acharya. He is a PhD scholar at University of South Carolina, Columbia, USA. He is also a teaching assistant at Department of Biological Sciences, University of South Carolina. His achievements include RMDL Graduate Fellowship Grant, CSIR National Eligibility Test in Lectureship with an All India rank of 41. And he had received summer research fellowships from Indian Academy of Sciences as well as Government of Karnataka VGS Division Group of Science and Technology Research Fellowship. And he has also published a lot of papers, technical reports, and is coming up with his books on butterflies of urban greeneries and butterflies of Bengaluru. He is also an alumnus of St. Joseph's College. He belonged to the CBZ batch of 2011 to 2014 and is an excellent speaker. Leave apart the technical glitches, you will hear a nice presentation today. So we will move on to Microsoft Teams for the presentation. Thank you.
Thank you, madam. We'll wait for that transformation to happen. Abhijit, you're telling me that the Bluetooth can work? Will that suffice the requirement of people on YouTube? Or yeah, that's what that is happening. Rachel is working on that. She will get the ID done. Hope Nitin can stay on. Abhijit, if you can confirm that we are not eating into his time. Busy schedule there as well. Okay, but we have to give the link. got our link. It will be now posted here on the chat box. Participants, the Microsoft team link is ready. Shortly, it will be posted here. You can all click that and join. Also, alternatively, the link is sent to your mail. Please check your mail as well. Participants, please keep checking your mail. You will receive the link there, and you'll also receive it here on the chat box. Volunteers, NSA office bearers, please post the same on the YouTube and the Insta links also. Okay, can you post that link here on the chat box so that people can access?
Hello, can you hear me now? Hello. 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 Oh yeah, everyone can hear me now? That's perfect. That's great. And this is also being okay. So do we wait for a few more minutes? Just a second. Yeah, I need to. Yeah, just give me a second. I do not have. What do you call? PowerPoint on my. Yeah. Windows partition. So let me just open.
Okay, Abhijit,
Google
is
Slides.
that posted here? Yeah. Okay.
How do I share screen here?
Abhijit, people are asking for YouTube link also.
Yeah, share. Okay, perfect. PowerPoint, screen.
All of you here, please join the Microsoft Teams. The link will also be shared on your mail. Please check. Please join us there. Sorry for the inconvenience. Thank you for bearing with us.
Can you hear me now? Okay. What about the others?
Yes, sir, we can hear you.
Okay, finally. Okay, so do we wait for a few more minutes or do I?
Nitin, can you hear me?
Yeah, I can hear you, sir. Yeah, I can hear you. Yeah.
Abhijit, can you hear me? Okay, okay. Nitin is also audible. Okay.
Yeah. I mean, I just don't understand what happened with Google Meet. I tried it on 6 different laptops, and none
Oh
of them
my
are
God.
working on Google Meet. I mean,
I'm really sorry. Sorry for that inconvenience. Thank you for bearing with us, Nitin.
No problem at all, sir. I mean, thank you for, I mean, I should also thank all the, participants for bearing with this, technical snafu. I mean, I'm so sorry for that.
No problem. I think we'll finish this.
Yeah, I mean, I'll try to rush a bit as well. So probably in the next 30 minutes or something, I'll try to finish it as much as possible. And
Exactly.
whatever.
No problem. If you have time, please start from beginning so that many of them missed it. That's the reason.
Yeah, I mean, I
You
don't
can
mind
go.
starting from the beginning. Yeah. So let me know when to start and I'll be ready to start.
Sure, sure.
Faster, no problem. Already you finished no half of it. No, Jay? Hello?
Abhijit, has the link being sent on email? Soumya has sent it. Okay, okay. How many participants? I am not able to use my mobile.
30 plus, Abhijit.
And you can see point, right?
I think many left it there. They should be here.
Can you see my screen?
Yes, yes, Nitin, it's visible.
Still 58 people are there, yeah.
Jai sir, I'm admitting everyone inside.
Please, please.
Yeah.
Yeah. I think people are also asking for link on the mail. Okay, Abhijit, shall we start then? We have around 57 people, participants.
That's a lot.
No, the rest will come through YouTube, I believe.
Do we give it 1 more minute and start?
Okay, okay, Nitin.
Yeah, I mean, or if you want me to start, I can start away.
That's what. The number is shooting from 57. It's gone to 64 because I need to log into this.
That is true. The Microsoft Teams.
Yeah. I think you can start with the introduction onwards.
Right.
As participants come, they can catch up with where you left.
Yeah. All right. So again, very sorry for the technical difficulties, and it was something was totally out of our control. So hopefully there are no more difficulties, and I will try to finish this as soon as possible. And if there's something which which I'm not clear with, or if I'm not audible, or if I'm not making sense, please shoot me a message, and I'll try to answer each and every question either after this presentation, and Jai Shankar sir can actually send it across to all the participants who ask questions, or I'll try to answer it if we have enough time in the end. So for today's talk, I will be presenting a short primer on understanding the ecology and evolution of moths. And before we got cut off, I spoke about a few things. I only spoke about what moths are and why. Can everyone else mute their audio? Yeah. I think someone still has their audio on.
Tanushri, please mute yourself.
All right. So yeah, I just touched upon a bit of what moths are and why moths make a very interesting study system, and how moths have diversified and evolved over time. So to give a very brief introduction, I'm not going to explain in detail. Moths belong to the order Lepidoptera, which means scaled winged insects. There are 180,000 species of moths. Like, many of them are undescribed, and we do not know anything except their name for at least almost like 90% of the moths. It's very, very understudied, very poorly known a group of insects. But whatever is known from a few of those moths, we know a lot more about them. For example, the silk moth and yucca moths and Plutella moths. And there are 120 families, and this keeps changing depending upon what types of classifications the taxonomists are following. And these are holometabolous insects. That means they undergo a complete metamorphosis, whereas grasshoppers or other things are not holometabolous, because they just increase in size whenever they are right after hatching out of their eggs. Whereas moths have a larval stages, 5 larval stages, then they have a cocoon stage, and then they emerge out as adults. That's why it is known as holometabolous. And females are heterochromatic in moths. That means females have X and Y chromosomes, whereas in humans, it's males who have X and Y chromosomes. And most moths are usually nocturnal, but many of them are diurnal or even crepuscular. And I'll talk about these, in detail in the coming slides. And moths have been used to study pheromone diversity in biology, because each moth has its own pheromone signature. And if you have at least 2 lakh species or around 2 lakh species, you have such a tremendous diversity of pheromones. And because moths can either be diurnal or nocturnal or crepuscular, you need to have fine tuning of vision to be able to see in those environments. So therefore, moths actually serve as a very, very great system to study how vision has evolved, what kind of structures are there in the vision, and how natural selection shapes the diversity that we see in visual genes. And hawk moths specifically have been used tremendously in flight dynamic studies to look at how moths fly and how do they turn, and how what kind of systems they have to enable fast flight and enable efficient flight, and even their bodies morphology to aid their flights. And coevolution is something that has been extensively studied in moths, and I'll talk about this a bit more in the later on in the presentation. And a really interesting thing about a particular species of moth known as Galleria melonella. It's one of the few moths which have been used to study toxicology, the effects of toxic compounds on humans, because this moth has the immune system similar to a human being. A lot of research has been done where they test a lot of compounds on this moth, see what the reaction is and how well it is tolerated and what the effects are on its body, and that has been translated into human medicine or any sort of toxicology studies. And Helicoverpa armigera is one of the many pests that are present in moths, and these are devastating pests which cause up to $40 or $30 billion of loss to agricultural crops every year. So this was the basic, background as to why moths are such good model systems. And then I spoke about the first fossilized, the earliest fossil that we have of a moth is known as Archaeolopus main, and this is very closely related to caddis flies. The body form is not, similar to the present day moths, but it kind of resembles the present day moths, and also it has wing venation, similar to present day moths. And moths kind of originated 300 million years ago. And I was specifically talking about what are some of the features that promoted diversification in moths. And the first one is when land plants evolved. That was one really important event which promoted the diversification of moths. And the second one was the evolution of proboscis, which gave them a special niche to feed on in terms of nectar, so they were able to diversify further. And the evolution of specific female mating and egg laying organs ensured that they even they could diversify even more. And one really interesting thing, when you look at the phylogeny, is that butterflies and macro moths evolved around the same time, and that was because it was right around like 60-65 million years ago is when the Earth kind of started warming up a bit more, and you could have larger body forms to actually evolve and sustain on Earth. So so most of the large-bodied animals are closer to the tropics, whereas if you see especially in insects, and if you see you kind of see smaller bodied insects as you go away from the equator. But if you look at it in terms of large mammals, it's the opposite. It's called as the Berg's law, so you can read about it when you have time. And one interesting thing that I forgot to mention is that this is the best phylogeny we have so far on moths, and this is only up to the super family level. I mean, we do not have a very good phylogeny when it comes to species level. I mean, forget about species level. I don't even think we'll have a good phylogeny of species level in the next 10 years. It is just so tremendous in diversity. Sequencing them is a huge task for any scientist, but who's studying moths. And then this is where I stop. So I'm going to continue at my regular speed from here. So if you look at, if you have observed moths for a long time, you will be thinking, okay, fine. Why are there so many geometrid moths, and why are there very few zygonid moths? So what is that, which makes one particular groups of moths more diverse compared to the other particular clade of, moths? And each group is known as a clade in biology, in phylogeny. So you can talk about, zygonid moths as a clade. You can talk about chaternid moths as a clade. You can talk about Erebidae moths as a clade. So each group is known as a clade. And this thought is actually not new. So the diagram here is from Darwin's book, which is, Origin of Species. Even when people mute their screen, please. I mean, their audio. Yeah. So when Darwin first started thinking about species or how species were related, he knew that a lot of species, there were some groups which had many species, and some groups which had very less species. So he started thinking about what could be causing that, and that is something we are still studying right now. Looking at what makes a particular group of species diversify more. And one of the best examples we have of what makes some groups diversify more and some groups less species rich is by studying silk moth phylogeny. This came out last year, and there's a really good phylogeny which provided a lot of information as to, I mean, this has been studied in a lot of other organisms as well, but this phylogeny is specifically to moths kind of gave us an idea as to why some moth species or moth families are very diverse, or what can be causing it. So to understand that, we need to understand three terminologies. The first one is speciation rate. So that is basically the rate at which new species form from existing species. And then the second one is diversification rate, and that is the difference between speciation rate and extinction rate. Extinction rate is when the rate at which animals die, and animals or plants or any other organism dies, and speciation rate is when new things are born. So diversification rate is where you subtract speciation rate from extinction rate, and then you see what the effect is. So when you do, I mean, you can do that using molecular evolution tools, and you can plot that on a phylogeny, and this is what they've done here. So when they did that specifically for the Bombicoidae superfamily, they found that there were some groups which had very low diversification rate. So if you see the legend here, so 9.1 and 19. So the redder it is, that means the fast, it has more diversification rate. That means it is accumulating more species compared to the number of species lost, and the bluer ones is where it's accumulating lesser amount of species as species go extinct. So Spingidae and Saturnidae have very high diversification rates. That means they are, you have more species than extinctions. Whereas, if you look at Brahmidae or other, Bombicidae moths, you have lesser species compared to what extinctions are. And what does this depend upon? So this actually depends upon three different, things. So the first one is environmental conditions. So if you have a very uniform kind of an environment, let's say if the entire world was a grassland, you would not have very diverse species because all of them would be competing for the same sort of resources. So you would have a lot of competition and you would have a lot of extinction. Whereas if you have very varied environmental conditions, like we have in India, the Western Ghats is completely different compared to what we have in the Deccan Plateau, compared to what we have in the plains of North India, compared to what we have in the Northeast, and compared to what we have in the Himalayas. So different kind of environments give rise to different kinds of adaptations. So therefore, so whenever a particular can someone mute it? That's can someone mute their audio, please? Yeah, thank you so much. Yeah. So where was I? Yeah. So what happens is that, let's say a moth which is found in the grassland is starting to adapt to evergreen forests. That means when it goes into the evergreen forest, there are not many moths which it is competing with. So it is kind of released from competition and it can start to specialize and diversify. So that's one of the reasons why you have diversification rates higher in some groups and lower in another groups. And so if you see, for example, Geometridae moths, you could say, okay, fine, are they found in various different types of environments? If they are, that means, okay, fine. If they are found in so many different environments, you can expect them to be more diverse compared to, let's say, an organism which is very, very restricted to in one small, hillock in the middle of nowhere. And the second thing which influences is the age of the branch. So for example, if you see this phylogeny here, whatever horizontal line you see is the branch, and you see some branches are short, some branches are long. So longer branches mean they evolved a lot earlier in time. That means they have been there for a longer time. So when you have an older branch, you have more chances of accumulating species over a period of time. Just as a fact of that, older branches usually have higher species richness in them. And the third and the most important thing, which kind of also, Darwin noticed, or probably the only thing one of the main things that Darwin noticed is population size. So when you have larger population sizes, whatever mutation or whatever effect you have on the population is kind of, nullified because it just gets lost in the population. But when you have very small populations, whatever smaller, mutations, whatever smaller adaptations you do see kind of get expressed and passed on to the next generations much, more compared to when the population is larger. So when you have smaller population sizes of a lot of individuals, then, you kind of start diversifying a lot more compared to a larger population. So if you can, whenever you do go out, think about it in terms of, if I'm seeing a Brahmidae moth, if I'm seeing Brahmia, how many individuals are there? Is it a larger population? Is it a smaller population? And then start looking at start thinking about these things. This will actually help you understand as to how species assemblages have evolved on Earth. So these are the three things which actually influence diversification rate. So now that we know, we have so many moths, we have so many, diverse families of moths, and this is how they diversify and this is how they specialize and all that. Let's go into actually learning about the life history traits of moths. So this is something I'm pretty sure everyone is familiar with, but I'll just run through it very briefly. So the life cycle of a moth starts with the female laying eggs. I mean, usually when you have an egg, it hatches directly, or it can go into a diapause if the conditions are unfavorable. And then when the conditions are favorable, the egg hatches, and you have 5, different instars in Lepidoptera. I mean, that's like a standard. You have 5 different instars, and once these 5 different instars are done, the moth caterpillar, spins a cocoon, and then it, encapsulates, in the cocoon, and then it flies out as an adult. So this is the life cycle, and each of these life cycles have been, each of this life stage has its own threats to, existence, own adaptation to survive, and own behavior. So I'll try to highlight a few of these in the next few slides. And please remember, whatever I do show is just a tiny portion of what is actually there out in nature. It's just tremendous, and we can talk for years about, what the life history evolution of moths are. So I'm giving you a very small opening window to it, and if you're interested, I can recommend, a lot of resources that you could use to understand these better. So the first thing that comes to anyone's mind, how does a female know what plant to lay eggs on? Or how does a female search a plant? I'm pretty sure all of us started out thinking, on these terms when we started learning or observing moths. So to go to actually explain the mechanical basis of this, I'm going to go through a very short pathway. So the first is, let's say a female lands on a host plant using olfactory and visual cues, and I'll talk about these cues in the next slide. But imagine, okay, fine, a female sees a host plant and she identifies it using olfactory, that means smelling, and visual, that means seeing. So she sees a plant and she smells it and she says, okay, fine, this is what smells like. This, smells like food to me, smells like food for my caterpillar. So she goes and lands on the plant. But that is just the first step of what happens in oviposition. And then what happens is that if you have observed what females do closely, is that whenever they go onto the plant or any surface that they think is fine, they start scratching with their forelegs. Yeah. So when they scratch with their forelegs, the plant breaks open, and it releases a lot of chemicals from the plant, and those are known as oviposition stimulants for the which oviposition stimulants basically which induce egg laying in moths. And these oviposition stimulants receptors, that means the things which can identify these oviposition stimulants, are actually in the legs of all these moths, in the forelegs, the first 2 legs. Basically what the moth is doing is basically it's tasting with its leg. So it goes there, it scratches, it tastes, and says, okay, fine, does this taste like my host plant? And there are specific receptors known as gustatory receptors. Gustatory receptors are those which give you the sense of taste. So in your mouth, if you have very less bitter gustatory receptors, you will not taste bitter. You might be able to eat bitter substances without a problem. So, the evolution of gustatory receptors is something which is very, very interesting, and that can, itself be talked about for days together. And there's also something that I'm working on in my PhD, but I'm not going to go in detail about the evolution of gustatory receptors. So, the receptors are specific to each stimulant. For example, let's say if a female goes and lands on a plant, she needs to be able to taste compound A, compound B, and compound C in the right concentration. If she does not get that taste, she will say, okay, this plant is not suitable for me, I'm going to find another plant. And if only A or B is present, she will still not identify it. And that is why, these gustatory receptors are very, very specific to, each and every, host plant range that the females can accept. And then what happens is that this, information is processed in the central nervous system, and that signal kind of, elicits, egg laying in females. So goes on to, briefly, just run through it. Female lands on the host plant, scratches the host plant, tastes the host plant, and the taste has to be very specific to what is acceptable to the female, and then that information is relayed directly, into the central nervous system, and then you have, egg laying occurring. And there are a lot of factors that affect host plant choice. The first one is temperature. So when you have lower temperature, first of all, the moths are not active in general, and even when they're active, their the processing capacity or the transmission between central nervous system and the gustatory receptors are low, so you kind of have lesser specificity to host plant. So the females will not be able to identify the host plants very well. So when you have the optimum temperature or at a higher temperature, females are able to identify the correct host plants. And host plant quality in general. Let's say let's say a slug moth lays its eggs on castor plant. So for it to actually lay egg on castor plants, the castor plant should actually have the desired quantity of resin in it, the desired quantity of other nutrients in it, and all of this is assessed, or it is kind of made a decision when the female starts scratching the leaf. So it can assess host plant quality, it can also assess plant chemistry, and then it can make a choice as to if I should lay eggs here or not. So host plant quality is something which can be affected by temperature, it can be affected by what kind of soil the plant has been growing and what kind of other stresses the plant has experienced previously. So host plant quality itself is a huge field. And the third thing is plant chemistry. Each moth species has its own set of plants that it can lay eggs on, and that is directly tied to what kind of gustatory receptors each of the moth species has. And the gustatory receptor evolution and host plant specificity is tied together. So you need to have the right plant, right kind of plant chemistry. For example, if a slug moth needs to lay egg on castor plant, it needs to be able to detect resin in the plant. And also when a female tries to go lay eggs on the plant, if there are larvae which are already present on the plant, the larvae actually start secreting chemicals through their feces, their poop, and that can actually prevent additional females to lay eggs. And that's kind of a strategy to prevent competition on a single plant. So if there are larvae already present on the plant, it's very unlikely that the female might lay eggs. And if she has no other choice, then she'll lay eggs. But if she has other choice, she'll kind of just move on to another plant. And the degree of specialization, that what I spoke about in terms of very narrow set of host plants for moths. And one really interesting thing about moths is that this is very, very fascinating with moths, that the gustatory receptors are very diverse in moths. So each some moths have like 30 or 40 different types of receptors. That means they can, if you do permutation and combination, they can actually identify that many plants, that many kinds of plants. Sorry. So that's why you have absolute generalists in moths. I mean, in terms of host plant ability to feed on host plants or lay eggs on host plants, moths are can be absolute generalists where they lay eggs on like hundreds of plants, or they can be absolute specialists where they can just lay eggs on one plant. So all the pests that we see, they have very diverse gustatory receptors, therefore they're very generalized and they can feed on a lot of things. And once the female lays the eggs, what are some things which affect how well a larvae develops? So the first thing is physical defenses. So for example, A, B, C, D, and E are different types of physical defenses that the plant actually has to prevent caterpillars from eating the plants. So A is thorns, B is modified leaves in cactus, C is modified stem in rose, D is the hairy substances that you see in a lot of plants and fruits, and E is very tough leaves. So all of these modifications kind of makes it hard for the caterpillars to feed on these plants. And you also have a lot of plants which deposit calcium oxalate crystals, so that will kind of either make it really hard for the caterpillar to eat or just poison the caterpillar. So those are some of the physical defenses that the plants have, that will make it harder for the caterpillars to feed on. And you also have a chemical defense, which is something which a lot of chemical ecologists study, a lot of plant insect interaction biologists study. So I'll try to explain this in a minute. Within a minute. So let's say you see a moth caterpillar, any sort of moth caterpillar munching on a plant. So what happens is that the plant has direct defenses where it kind of sends in all its chemical compounds to the leaf, makes it harder for the plant, caterpillar to eat. But the caterpillar has specific gustatory receptors which it can actually overcome these direct defenses, and it kind of eats it very well. And then what the plant does is it kind of attracts predators using chemical signals, and you have ants, you have parasitoids coming in, and they will start attacking the caterpillars. And that is a kind of indirect defense. But what do caterpillars do in, when that happens is basically they have various adaptations. For example, slug moth caterpillars have spines on them. A lot of, most of the moth caterpillars build, they roll the leaves and, hide themselves. So all of these kind of prevent parasitization or predation. You still see a lot of parasitization and predation, but if these caterpillars did not have these defenses, they would, all the moths would have gone extinct. So this is an overview of how plant defense and caterpillar, counter defense occur, and how these two things can actually keep evolving over a period of time. So each of them keep updating, upgrading their weapons, and this is an never-ending race. And this is actually known as evolutionary arms race. So you can actually read about it in any sort of ecological literature. Prey-predator interactions, herbivore, plant interactions, and pathogen and host interaction. All of these are evolutionary arms race, where both of these things want to stay alive, and they keep upgrading their arsenal of weapons, and they keep fighting out this battle all the time. And when I told you guys about generalist versus specialist species of caterpillars, this has a lot to do with what the host plant chemistry is. So here are 4 caterpillars. This is a study done in 2008 by Roslin and others. So the first one is 1, 2, 3, 4 is 1, 2, 3, and 4. I don't want to spend more time on the species names. So what they did here was 2 of these are specialist moths which can feed on Vescalagen. 2 of these moths are generalist species which cannot feed on Vescalagen. So generalist, that means they can feed on the plant, but they're not very well specialized to feed on Vescalagen. So what they did was they put all of these 4 moths on plants, and they added on substrates, and added either tannins, which are secondary compounds present in most plants. So they wanted to see how it happens when you have a common denominator kind of a secondary compound, and they added specific Vescalagen, which is specific to the plants that these specialized caterpillars have evolved on, and some of these generalist caterpillars can also feed on. So what they found out was that the amount of leaf that was eaten was also very less between these 2 species, which are generalist. The amount of body mass gain was also different when it was when Vescalagen was added for these generalist species. So what this kind of tells us is that specialized species can actually feed better when there is secondary compounds, when there is toxic compounds, whereas generalist species which feed on a lot of plants can still feed on it, but their quality of life will not be that great. So for example, if you think of Helicoverpa armigera, which feeds on 400 different plants, which can feed on toxic tobacco plants, but it's in terms of its growth, in terms of its weight, in terms of its overall health condition, it will not be as great as, let's say, a tobacco moth, right? Tobacco moth is specifically evolved to feed on tobacco. It can do very well. It can outcompete Helicoverpa, and that is how these evolutionary dynamics play into picture, where you have specialist species which are exploiting these plant resources very well, and you have these generalist moth species which are able to exploit a lot more plants but not as effectively. And both these strategies have pluses and minuses depending upon what context you're talking about. So now that we know how moths lay eggs, how moths locate mates, let's how moth lay eggs and how moth caterpillars feed. Let's talk about how do actually moths locate their conspecific mates. And the most important and the most specific things, most interesting thing about moths is pheromones. So you have these 2 moths here. You can see this moth has its, all its secondary sexual characters, averted out, and it's, when it does this, it is sending out a lot of specific chemicals out in the environment for the females to sense, and also the females do the same thing. Moths are one of the few groups of animals where both males and females have specific pheromones. And this is something which is not seen in butterflies, where males have most of the pheromones, whereas in moths, a lot of males and females also have it. And this is another, modified structure where, it is sending out chemical signals. So what are the qualities of pheromones? They are very highly species specific. So this happens because they do not want to mate with species which are not their own. So it kind of prevents hybridization, because whenever you have hybrids forming in nature, they're usually sterile, and there's no point of, you really cannot reproduce after the first generation. So hybrids are usually not favorable, and therefore these pheromones are very highly species specific. And another characteristic is that they have very long range communication. I mean, you put out a specific type of pheromone out in the field, you can attract moths in no time. I mean, it can get moths from kilometers away, because these chemicals are volatile, but they do not dissipate. So you have these whole, molecules of these pheromones traveling for very long distance. And that is something chemical ecologists have been studying for a long time, and there are very, very interesting results from it. And these pheromones are also under intense natural and sexual selection, because the females can only mate with those males which have very high quality and very strong signal. So over a period of time, you only have, moths which have very high quality pheromones and very high, intense pheromones. So it is always under selection. It is always under intense selection. So you have that, that's why you have specialization of very specific compounds. And that's why you see such a huge diversity. And like I said, moths are in one of the very few groups where you have both the sexes having pheromone present in both the sexes. And this is something which is very, very interesting. So there is actually a database known as the PheroBase, where people have been depositing all the pheromones that they have found out, all the pheromone in the chemical structure, the NMR structure, everything that is known about the pheromone. And they have more than, I think, 30,000 entries, and it's crazy. It's if you really want to study pheromones, if you want to study how pheromones are related, if you want to, you can use these pheromones to actually plot on a phylogenetic tree to see how pheromones have actually evolved in nature. You can use this database. You can go there. You can download whatever pheromones you're interested in, whatever moth species you're interested in, and then look at it. So this is pherobase.com. If you have time, if you're really interested in looking at pheromone evolution, this is something I definitely recommend you guys to check out. So that is the basic life history of moths. And now the next thing is, what do moths do? What kind of behavior do moths have? And what is the importance of having moths in nature? And one of the most, the two most important ecosystem services that moths provide are, one is they are prey for a lot of predators, and two, they are pollinators. And people might, a lot of people thought moths might not be efficient pollinators, because whenever they usually studied bees, and bees have way more pollination capacity compared to moths. Totally, agreeable. I do agree with that point. But one thing where moths outshine any other organism is nocturnal pollination, because none of the other insects are active during the night. Very few insects are actually active during the night, especially proboscis, insects with proboscis, and moths are actually probably the only ones which are active at night. So a lot of flowers, a lot of plants have evolved in tandem with moths to be able to specifically give out signals to be pollinated in the night. So for example, let's say if you see any night blooming flower, you know that A, it is usually white in color, and B, it is very, very fragrant. I can say fragrant, or it can be, it has a very strong odor. It can be pleasant odor, it can be like a corpse, it can be like feces, it can be anything. So a lot of night blooming plants have specific adaptations which actually have evolved to help moths locate them easier and help them pollinate better. And why, you can ask me the question as to why did plants evolve to become pollinate in the night? Because in the morning there's so many, during the daytime there's so many plants which bloom, and there's so many insects competing for nectar resources. So a lot of plants might actually not get pollinated, and they might not reproduce. So shifting to nocturnal pollination was beneficial for plants as well as hawk moths, as well as moths, because the moths didn't have to compete with other insects in the morning. So that's a background about why moths and are important nocturnal pollinators. So when Darwin actually went to Madagascar, or when he got specimens from Madagascar, so he saw this plant here. It's an orchid known as the star orchid. He was very surprised with the flower. The flower is probably 4 or 5 centimeters in length, but it had a 30 centimeter or something long corolla tube. The minute he saw it, he said, I wonder what kind of a moth could be pollinating it. He knew it was a moth pollinating it because this bloomed in the night and this had very fragrant smells. But Darwin never saw the moth. Darwin didn't even know that if a moth like that ever existed, or was it just him hypothesizing, saying that, there could be a moth actually pollinating it. But after 150 years or 160 years, scientists actually found the moth in Madagascar. And when they found it, they were not surprised because the moth had a 30 centimeter long proboscis. This is known as the Darwin's moth. So, this flower can only be pollinated by the Darwin's moth because, to reach the pollen, it has to go all the way to the end of the tube, and no other moth can, enter that distance except this moth. So a lot of these moths and, flowers have co-evolved so tightly that without the moth, the plant cannot survive, and without the plant, the moth cannot survive. So this has a lot of implications in habitat, destruction, because if you're destroying even a part of a small habitat where this plant could be available, it has a very strong implications on what it could do to the population of these Darwin moths. And this is something that we should always probably worry about, because in India, we have so much destruction of habitats going on. And people might say it might be a grassland which is getting destroyed, it might be a wasteland which is getting destroyed, but each and every, habitat has its own set of locally adapted and co-evolved interactions. And destroying one also destroys the interaction. So it is very important to keep in mind, or start thinking about these issues. So this is, there is not a better example of textbook level, example of co-evolution apart, when you compare the Darwin's moth and the star orchid. This is literally the best example one can give, and that comes from a moth. And another thing which has been studied extensively for the past 50 years is the yucca plant and the yucca moth. So yucca is a type of desert plant which is found all over North America. And what happens here is that this moth is an obligate, mutualist with the flower. So when I say obligate, that means it has no other choice but to just depend on the flower. So what it does is, the female, the moths usually pupate in the ground, and once they emerge out of the ground, they mate on the plant. The males, arrive on the plant, the females arrive on the plant, and they mate on the plant, especially on the flower. And once mating is done, the female goes into the flower, collects the pollen, and then flies out to another flower. And then, when she goes out to another flower, she goes into the developing ovules and then lays her eggs on the ovules. And the larvae actually, start eating the seeds, but in the process, she's also pollinated the other flower. So without, the yucca moth, the yucca plant will not survive. And without the yucca plant, the yucca moth will not survive. It's literally the most specialized co-evolved interaction there is in terms of pollination. And people have been studying this for 50 years. Studying this moth has given us insights on co-evolutionary processes. It has given us insights on local adaptation. It has helped us understand how is that, such a specialized interaction can evolve, and what are the consequences of these interactions, and what does this help us understand, how will this help us understand the biology of moths in general. So it is a very, very interesting system to study pollination. There is not a better system than moths to study pollination. And there is also other moths, for example, the Plutella moth, which feeds on another plant, which has another obligate mutualism with it. So you can find tons and tons of examples. Even in India, we do not know of it yet. That is because we have not been looking at moths that closely. So in terms of, if I have time, I'll probably spend 3 or 4 minutes kind of discussing a few student projects that people can do just by staying in your backyard, or just by, visiting your local park. So hopefully we will have 5 minutes at the end. I'll try to rush in a bit more. And when you have, pollination occurring between, moths and plants, there's, you kind of have co-evolution happening. So the moths evolve specific characters to be able to feed on those specific flowers, and the flowers have, specific adaptations to help the moths feed better. So and the way we study these interactions is through pollination networks. So here is 3 pollination networks. The first one is between moths on the left side, 103 species of moths with pollinating 47 species of plants. And this one is hoverflies and butterflies on the left and the plants on the right. And this one is moths. This one, the orange one is the bees and wasps on the plants. But if you see here, moths have the highest diversity of pollination networks compared to any other species. Even if you group in butterflies and hoverflies, you still have 76 species in this landscape. Whereas when you just study moths, you have over 100 species. What this says is that moths are very, very co-evolved to, in terms of pollination networks and how much they can pollinate. So diversity of moths is a very, very good indicator of plant diversity, a very good indicator of biodiversity in the region. You can have, a lot more bees and butterflies, but moths actually can indicate how good or how specialized or how diverse the landscape is. And we usually do this by, doing field surveys, and we analyze pollen of proboscis. So what we do is we collect moths and we scrape the pollen off their proboscis using a small pen. You do not damage the moth, and then you release the moth. And then you study the pollen under slides, and you have guides which says what pollen belongs to what plant, and you can actually construct these networks. And this is something which a lot of students can do back in India, where all you have to do is just set up moth traps, and using whatever required permissions and required skills you need to develop. Once you develop those, you can briefly handle the moth. You can use a pen, spread the proboscis and then collect the pollen and then release the moth, and then use those pins and mount a slide using all of these pollen grains, and then take the help of the amazing botany professors that we have all over India, and then identify all these pollen, and then you can build these networks, and you can actually have a better understanding of what kind of system services do these moths provide. So that's a brief overview of the behavior of moths. And last thing that one thing that I want to touch up on is the anti-predator adaptation in moths. Because moths are herbivorous insects, they are preyed upon left, right, and center by a lot of organisms. So moths have very specific anti-predator adaptations which help them survive in this world. And the most well-known of that is echolocating. Probably the most common predator of moths are bats, because bats are active in the night and moths are also active in the night. So what happens is that bats kind of eat moths all the time. So how do moths avoid bats? So we all know that bats use echolocation to identify moths. So what moths have developed is that they use their hind wing tails. So if you see here the tail here, to jam the echolocating signal that the bat is using and just escape from there. So people did a bunch of experiments. They're still doing a bunch of experiments to look at what was the effect of tails on survival. So this is the capture rate. That means how many percentage of times were the moths captured. So the moon moths, which had their tails intact, were captured at a very low rate, like around 30%. Whereas if you remove the tails of if you remove the hind wing tail of the moths were much more captured frequently. And they used another moth as a control. It almost had 100% moth. 100% capture rate. Those moths did not have any chance of surviving the bat attack. And they also compared it with another moth which is not present in the area, and even that had an intermediate level between intact and cut off hind wing tails of this moon moth. So these tails actually help in jamming the signals, because they start beating rapidly when they sense these echolocating signals from bats, and they move away and they jam it. So bats really cannot locate. It's like basically blinding a bat and asking it to find you. So it has a very low chance of finding it when your tail is intact, and it will definitely catch you when you do not have a tail. And another thing with moths is that a lot of moths are aposematic. Aposematic is where you are toxic and you also exhibit it. So poisonous organisms or toxic organisms usually have very, very bright coloration, so that predators kind of remember it and do not attack it at all. So there are a lot of moths, for example, zygonid moths and a lot of tiger moths, which are aposematic, which are very toxic, which are also very, very colorful, which have very bright colors and patterns. So if you have to develop that amount of toxicity, if you have to be able to, have adaptations to develop such toxicity, you need to lose something, or you need to have a trade-off, because you cannot have the best of everything, because it's very costly in nature. So there is always a core. What people have found out was that if whenever you go out in nature, if you see something which is flying very lazily or nonchalantly, very lazily, you can kind of get make an assumption that it is a toxic moth. Because for example, let's say here in this graph, in the first part of the graph, from right to left is how fast the flight was. So this is very fast flight. More gray means more fast flight, less gray means it's very slow flight. So the one here on the right is the slowest flying moth, the one here is the fastest flying moth in the study that they did. And what they found out was that the more black you see, the more toxic they are. So moths which were highly toxic had very, very, very slow flight. So that means they are showing their flight. They are saying, I'm lazy because I'm not lazy. So that's a very bad way to put it. My flight is lazy because I do not really need to invest any energy in flight, because I've already invested all the energy that I need for protection in aposematism. So that is the trade-off you have. When you have no when you do not have any toxic compounds to protect you, you fly fast because you need to run. Either you run or you are toxic. So that's the trade-off that exists. And this is very, very well studied in moths, and tiger moths and zygonid moths are absolute model systems to study these. And another thing is that, bats have, not bats, moths have also evolved ultrasonic hearing. So that means, because bats use ultrasonic waves to detect moths, these moths have also evolved ears to detect them. And when people found out, how much time do moths invest flying in the night, they found that moths that had ears flew a lot longer compared to moths that did not have ears. So there's also a trade-off here. Moths that have evolved ears can actually spend more time flying and finding resources, whereas moths that have no ears, they have to be very quick in going about their life history, otherwise they will get eaten up. And this ultrasonic hearing is actually in an evolutionary arms race both in bats and moths, because bats are developing, they are evolving mechanisms where they can actually locate moths better, and moths are evolving adaptations where they can hear even the slightest of ultrasonic sound that they can perceive. So this is again under very constant selection, and they keep evolving more and more over time. And the last thing is with caterpillars. If you have ever touched the slug moth caterpillar, I'm pretty sure you would be itching for a while. And when people wanted to understand why do so many slug moths have spines in them and how did it evolve, so what they did was they used a phylogeny of the slug moths, and they plotted what character the slug moth had. Character is where you either have a spine or you do not have a spine. For example, here is the character state. So red is where you have spines present, and orange is where you have spines present initially and then you lose it, and white is where you have no spines at all, and then you have other states which are not important. So what they found out was that the spines evolved very early on in these moths. That means the ancestor of all of these slug moths had spines, and that kind of tells us that these guys have had predatory pressures from way back in time, and that's why they had to evolve it. And majority of the slug moths still have it, but there are few slug moths which have lost it completely. They do not have spines and they no longer sting. And when they actually investigated as to why that has happened, they found out that these slug moth caterpillars were very small, so having spines was actually counterproductive, because the spines would have to be bigger enough, where the spines had to be big enough to be effective, and if they had to be big enough, then they would have lost a lot more adaptations that they had. So smaller slug moths lost their spines, and slug moths which had lower predatory pressures over a period of time also lost their spines. So the white ones are actually in Australia, where predatory pressures in islands are different compared to mainland. So combination of small size and no predators kind of they lost their spines because of that. And lastly, I want to very briefly touch up on this, and I'll try to finish it in the next 1 minute. So evolution of vision in moths. Moths are the perfect system to study vision because they have such diverse, habit. I mean, their day activity. They're active during the day. They're active during the night. They're active during a lot of times. And to be able to see properly during those, you need to have very specific visual pigments, visual genes, and everything to do with vision. You need to be very fine-tuned. So when people started looking at what happens, how the evolution of these genes have occurred, they found this. So the basic unit of a visual gene is known as an opsin gene. So opsin is the one which is expressed when light hits, your receptor. And this one is actually by an Indian, Yash Sondhi, who is a friend of mine and who's also a very active, moderator and member of Moths of India. So he has recently published this. So he compared the gene expression of a lot of moths, which are nocturnal and diurnal. So the first one is just comparing 14 moths, and the second one is comparing how the opsin gene itself looks like, and the third one is looking at how it looks like when you superimpose it on the species itself. So what he found out was that there is a lot of differences between the diversity of opsin genes in diurnal moths versus nocturnal moths, and how much of expression it has over what kind of a vision you have. And that is really important for us because this is one of the few studies which says that there is differences in nocturnal vision and diurnal vision, and therefore you have that separation. And what another group of people also found out was that when you look at differences between male and female moth vision, you see a very significant pattern as well. So for example, females have higher red so on the x-axis is the number of days after you eclose, and on the y-axis is your expression levels. So females have higher expression on day one, when they first eclose, because most of their mating occurs the minute they are out. Because it's very rare to find an unmated female in nature. No matter how fresh she is, there's a very high chance that she is mated. So usually red is something that is used in mate location. So they have very high levels of red opsins on day one. And males have different kind of opsin patterns where they have more blue compared to the females, and they also have higher UV because that kind of helps in mate selection. And the levels of these opsins also change depending upon if you were in the dark or not. If you were in the dark, then you had a longer, you had higher expression of red, but your UV was not affected if you were in the dark or not. So all of these puzzles and pieces of puzzles kind of help us understand how did vision evolve and what kind of factors influence vision, and what do we learn from these, and what does it tell us about the world. So moths serve as the best example for it. So this is how opsin genes have evolved. So this is where my talk ends, and I'm really sorry for all the technical difficulties that happened. And something that I definitely recommend all of you guys to do is to contribute your records to iNaturalist and Moths of India, because these two websites are very, very useful for everyone in terms of citizen scientists, in terms of professional scientists, in terms of everyone. I mean, I have used iNaturalist so much, and my PhD would not have gone this smoothly without iNaturalist, because when I moved to the US, I did not know anything about the plants here. I did not know anything about where these plants were found or these butterflies were found. And it was because of iNaturalist that I was able to get all of these things much quicker than me finding it out by myself. So try to update all your records and everything, or upload your records here. And in terms of studying moths, I told you, some of the student projects that you can do is start looking at pollinator networks, because that is something which is not at all studied in India. It's very, very less. And we have such tremendous diversity of plants. We have such tremendous diversity of moths, and we can produce data and studies which no other country has ever produced. And we do not need professional scientists to do this. We need students, we need citizen scientists, we need people who are just interested, who are out there, doing fieldwork, who are out there observing moths. I mean, you do need, professional scientists to help you analyze data, but that's not a big deal. But you need people who are interested to collect these information about, studying pollinator moths and, pollination networks, or even observing life cycles, because we do not know the life cycles of a lot of moths, and that will actually tell us how specialized each assemblages and how generalized each assemblages of moths in the nature. And I will be open to any sort of discussions that you want in terms of planning your study, or if you want me to help you plan studies. I've passed my email to Tressa and Jaishankar sir. You can contact me anytime, and I'll be more than happy to help you all.
Thank you, Nitin. Thanks a lot for sharing with us a wonderful, informative talk on moth ecology and evolution. The session is now open for discussion. Dr. John Paul will take over. He will moderate the session. There are some questions here as well as on the YouTube link. So, Dr. John Paul, over to you.
Okay, Nitin, that was.
Okay, please speak a little louder, sir. I can't hear you. Yeah.
Can you hear me now?
Yeah, I can hear you, sir. Thank you.
Yeah. That was very much informative. It was really wonderful. Actually, after these many years, I myself I'm listening to such a wonderful, informative talk. Okay, that was very nice. Dear participants, in case we have any doubts, queries, in person you can ask Nitin now directly, or some people have posted in your in the YouTube link, as well as you raise your questions to the organizer. Let me read it out, not an issue. In case if you want to ask Nitin directly, you can proceed. Sorry, I have a power cut in my house, so you may not be able to see me.
Hi Nitin, Harsha here. I have a small question.
Yeah, so hey.
Does memory of the host plant as larvae play a role in adult choice? Will a moth that ate plant X as a larvae be predisposed to be laying eggs on a plant X again?
So that's actually a very interesting question, and that's something that I'm also working on in my PhD. But let me, I'm actually, I'll be able to answer your question a little better now. So a lot of people thought that, larval experience influences adult oviposition as well. So there were a lot of papers which came out in the 80s and 90s which said, that there is a memory of the larvae which feeds on a particular set of host plants, that its gustatory receptors evolve specifically to that, and moths will actually moths will lay eggs on that when they're adults. But that is not true in most of the cases, because most of the moths have gustatory receptors which are not that fine-tuned when they're larvae. So the gustatory receptors that are there in larvae is not correlated with the gustatory receptors that are there in the adults. And the only thing that you can be certain about what happens in larval stage is what kind of genetic background the female has in terms of being able to successfully transmit it to the offsprings, and they would be able to eat it. But there is not a lot of transmission from larvae to adults. And there was this one really interesting paper which is known as the title goes like this. It's called "Do You Know the First Time?" which is a very bad innuendo, but yeah, it spoke about larval memory and adult oviposition choice, and they found zero evidence for it. And it is possible in a few instances. For example, when you have host races, and that is there in a lot of aphids, a lot of grasshoppers, where you have over a period of time you have adaptation to a single host plant, and you cannot just go away from it. And that kind of evolves from a feedback between larvae and adults. But in moths and butterflies, you do not see this correlation. There is a very diverse set of gustatory receptors, and they are kind of not so tightly linked between larvae and adults. Did that answer your question?
Yeah, cool. I had one quick question, if I'm allowed to ask another.
Yeah.
So can you speculate why some certain species of very tiny moths, which are very weak flyers, are distributed very widely geographically? Like they're found on multiple different continents, say for example, the family Crambidae.
Yeah. So again, there are two ways where you can approach this. One is to look at when did Crambidae first evolve and where did they evolve. So for example, if Cramidae is a very old lineage of moths, you would expect them to have diversified a lot more over a period of time. And depending upon where they evolved, if they had evolved, let's say when it was still Gondwana, or it was just, when all the continents were still split, joined together, you could expect them to be distributed all around that continent. And even after the continent split up, they could diversify in their own continents. That's what happened to a lot of lizards, where that's how you have multiple diversification occurring in different continents. So in terms of Crambidae, having a small size is something. Smaller size actually is shown to have positive speciation rates. And that's because the energy invested in small size has, when you have smaller size, you also have very faster life cycles, right? That is kind of correlated. So when you have faster life cycles, when your body is small, you have more generations per year or per season compared to any other bigger moths. So once you have that, you can kind of expect more mutations to accumulate, more adaptations to accumulate, and that's why you could diversify more. So these are speculations, but again, this is actually a very interesting thing that anyone in the audience could actually, plan to study, because all you need is go to iNaturalist or Moths of India or any other website that has, species of Crambidae listed, take it down, and then, go to the NCBI website and download all the sequences for those moths and build a phylogenetic tree using software. And then you can, kind of see, okay, when did these guys come in and what kind of places have higher diversification rates. You can actually test this. But if you ask me, I think, that's my answer, that smaller size has faster faster life cycles. And depending upon how old the branch was, you could have more diverse nature of Crambidae moths.
Nitin?
Yeah.
This is Shiv Kumar here.
Hello, sir. How are you?
Fine. How are you?
Good, sir. Yeah.
Yeah. Today is one of the happiest days. Today I am asking you the doubt and you have to teach me now. Okay. You are just speaking about the OV deposition in moths on the plants, isn't it?
Yeah.
So as you answered just now to Harsha's question, do you think then the OV deposition every time will be a trial and error method for the moth?
Yes, sir. It is. I think all oviposition trials are trial and error, and that actually has a benefit for the moth. So moths which have very high specificity of host plant choice, where they do not make any mistakes, they do not evolve adaptations to feed on or lay eggs on other plants. So lot of generalist moths, they lay eggs on anything. They lay eggs on walls, they lay eggs on bicycle racks, they lay eggs on wherever they find. And that's because they do a lot of trial and error. So over a period of time, you kind of evolve, diverse receptors to be able to, identify more plants. So trial and error is actually a beneficial strategy if you are staying in the tropics. But if you're in the temperate regions, it's a very bad thing, because you first of all have very small amount of plants. You have probably like 3 months in a year where you can survive. And if you make mistakes, then there's a high chance that you will, go extinct.
No, that's a good idea. But trial and error method, don't you think the moths will have to invest a lot of energy going in search of a comfortable zone for OV deposition then?
So in terms of trial and error, it is not the female that has to, actively invest too much energy. It is the larvae which have to invest a lot more energy in searching the right host plant. So for example, imagine a scenario where there is a host plant in the region, but the female lays the eggs on some other plant. So the larvae will have to have enough energy or enough, capacity to actually realize that they are on the wrong plant and then start moving on to the right plant. And sometimes, this is possible when females have, if for example, let's say if a female was very healthy and if the male which mated with the female gave a lot of nutrients during mating, the females have excess nutrients, and this nutrients is translated into eggs. So the eggs are, fat rich, they're protein rich. And once the caterpillars hatch out and they feed on the eggshell, they have enough energy to move around. And depending upon, how wealthy the female was, the female can take a chance about trial and error. And you could expect at least, out of 100, 5 or 6 actually do go in and find the right plant. And my PhD specifically focuses on this because the butterfly that I'm working with, lays eggs on a plant which it is not supposed to lay eggs on, and the caterpillars are very weak to even move out of the plant. So all of the all of the caterpillars die, and we have seen almost like 20% decline in their population since the past 40 years. So it all depends on the life history of the moths and life history of the individual itself. But yeah, trial and error can be beneficial given certain circumstances.
No, but trial and error method on the flip side of the coin can also give a lot of adaptability in selecting the host plants, right?
Yeah. I mean, for example, if you have a female which is okay, let's consider two females where one female has very high specificity where she does not make any mistakes. She has a higher chance of laying her eggs on the right plant, so all her offsprings develop and they move on to the next generation. Whereas if you have a female who keeps doing a lot of trial and error method, she might not get a chance to pass on her genes. So over a period of time, you have moths which are adapted to very specific host plants as well. So when you look at it as a whole picture, trial and error is bad and good depending upon the context. And like you said, it definitely helps in adaptation over a period of time.
Thank you, Nitin. Nice to hear you.
Anybody else?
Hello?
Anyone else wants to
Hello,
ask
sir, I'm
questions?
Soundarajan. Hello?
Yeah.
Hello, sir, I'm Soundarajan from Tamil Nadu Agriculture University.
1 minute, sir. Soundarajan, sir, 1 minute. Nitin,
Yes.
I just want to know the time availability because we have extended it. No problem with you. You continuing to answer. Can you tell us the time?
For another half an hour or something. I go to the lab around like 11 a.m. So I still have an hour to go. So if I can stay on for another half an hour.
Yeah. Okay. Then, sir, proceed. Then I have a question. We'll take. Okay. Yes, sir. Please proceed, sir.
Yes, sir. Sir, Nitin, sir, your presentation was excellent, sir. Very informative. I have one basic doubt, sir. Regarding co-evolution.
Yeah.
Sir, you are discussing about the co-evolution between insect, particularly moths and plants.
Yeah.
So in the adaptation, if you see the insects or moths, they are having very shorter life period. For example, around 1 to 1.5 months, 30 to 48 days, the life cycle will be completed. Whereas in case of plants, if it is an annual plant, it will take more than 3 or 4 months. If it is a perennial crop, it is more than a year or more than 5 to 10 years also perennial crops are there. So between these entities, these two entities, the insects are adapting very faster than the plants. I think so. So can you throw some light how this phase of evolution between plant and insect, how it is coinciding, plants and insects?
Yeah, so that's actually a really good question because it kind of gets us to the question as to which came first. Did plants evolve first, or did insects evolve first, or did they both evolve together? And that's a very interesting question to ask, and that's something that has been studied for almost since 1964. So the first hypothesis what people put out was that plants, land plants evolved first, and then the insects started specializing on them. And so it was basically like insects tracking plant resources to begin with. And then once insects started attacking plants, the plants then started having defenses, and then the co-evolution arms race started. So you spoke about insects having shorter generations, but plants having longer generations. But if you think about it, a lot of these insects, when they have, let's say, 3 months, especially we'll talk about tropics and temperate region differently because they have different mechanisms. In the tropics, you have moths coming out year round. There are a few moths which are very specific to some months, probably in summer or in the winter, but you have a lot of moths which are perennial as well. They die and they keep reproducing. So in terms of plants evolving, it's mostly that they have very specific set of genes. So the one is the P453, the cytochrome genes, and a lot of basic secondary metabolite genes. And all of these secondary metabolite genes, they evolved a very long time ago. For example, let's consider mustard plants. When you eat mustard, you have this very pungent, you have a very particular smell. When you cook something with mustard oil, you can say it is mustard oil. So the mustard oil has specific compounds which evolved a long time ago. So what is happening now is basically the plant, let's say, the moth lays eggs on, this let's say it lays eggs in June or July, and then it completes its life cycle by September. So what happens by then is that the plants actually start upregulating its those already evolved systems, and they start having different combinations. Let's say if the plant has 10 different combinations, if when it first got attacked, if only 5 were active, it'll start getting the 6th or 7th compound active. So that kind of that small tinkering can make it harder for the uh, insect to feed on, and that those kind of tinkering capacities can evolve very quickly even in plants, because let's say you have a plant which reproduces every 10 years, so over a period of 10 years it has kind of accumulated a lot of adaptation in terms of diversity of its what do you call chemical compounds. And when you have the seeds, you have seeds which have all of these chemical compounds as well. So within the plant's lifetime and across plant's lifetime, you can kind of see both insect and plant coevolution kind of matching out.
Okay, sir. Okay, sir. Thank you, sir.
Nitin.
Yeah.
Jaishankar here. There are 2 questions from Father Melvin. Father Melvin was our rector here, and Father is from the botany department. He's a botanist. The questions are: will the absence of specific host plant cause end of the life cycle of the particular moth? 1. 2. Can the toxins present in this colorful moths he's referring to could be utilized for any useful bioproducts? These are 2 questions from Father Melvin.
Jaishankar, repeat once again the questions.
1. Will the absence of specific host plant cause end of the life cycle of that particular moth? 2. Can the toxins present in colorful moths could be utilized for any useful bioproducts?
Yeah, Father Melvin, that's actually 2 really interesting questions. The first one being, what happens if the host plant is gone? So that again depends on the degree of specialization the moth has. So for example, let's say you're talking about a generalist species. If 5 or 6 of its host plant goes from the habitat, it can still find another plant because it kind of has the mechanisms to combine different kind of receptors and still be able to taste plants and survive on it. But if you're talking about a specialized moth where it feeds on a very narrow set of plants, it is bound to go extinct unless there is a rapid adaptation to feed on some other plant. And there are instances where where we have seen rapid adaptations over one generation. And that is very, very specific examples. But yeah, in general, if you ask me, if the moths are generalists, then they will not have a lot of issues with plants going extinct. But if the moths are specialists, then they will definitely have a higher probability of going extinct. And that again brings us to the whole point of conservation biology, where you want to conserve species which are specialized. You do not want to invest too many resources on conserving generalist species, because once you have these specialist species doing well, you kind of know that you are also providing habitats and plants for the specialists, for the generalist species. So it is very important for us to make sure that we kind of know what these specialized interactions are, what these specialized plant combinations are, so that we can kind of make sure that the moths and plants don't go extinct. And unfortunately, we do not have this information for a lot of moth species in India. And that is something which I think a lot of students, I started rearing butterflies and moths when I was in first year BSc. Shiv Kumar sir, and Thomas sir can actually remember, me bringing in moths and cats and keep collect, when I used to just collect them and keep them in, all places in the, zoology lab. So it's like, this is something that students can actually start doing that now, so that we have more information for this and we can actually form better conservation efforts to, preserve these things. And the second question, where you spoke about these specialized secondary compounds which could be used for medicinal purposes, I think a lot of people are investigating this, and we have found a lot of these compounds are in fact very highly medicinal in nature. For example, the tiger moths, which feed on their host plants, they accumulate cardiac glycosides. So studying these cardiac glycosides have actually shown us that these things can help in contracting heart, or helping in heart, the health of the heart in a lot of human beings, because cardiac glycosides can contract the heart, or they can influence how fast and how the heart rate is. And studying these moths have actually helped us understand how the cardiac glycosides act, and we can actually use these specific cardiac glycosides which are in the moths to and prepare them synthetically and use them as therapeutic purposes. And there are also another set of compounds known as iridoid glycosides. They are again very helpful in using for a lot of blood disorders, and a lot of moths feed on it. And actually, by studying a few of these moths, they found out that the way the moths store these are actually in a very non-toxic form in their body. But when you break it and when you combine it with your saliva, with where the predators do it, then it becomes toxic. So a lot of these compartmentalizations and the way the chemical structure of these of the way the moths store it, we have learned a lot from it. And I think in terms of we have prepared those synthetically in the lab, but in terms of actively using it in medicine, I think we are still a bit away because of all the issues involved with human trials and things like that.
Any more questions?
Dr. John Paul, if there are questions, you can put up.
Yeah, there are certain questions raised from the YouTube link.
Yeah,
can I read it?
Yeah, please read it.
There are a few questions raised by Krinmi Nagbant. The first one is, are we using the ultrasonic waves created by bats for abandoning moths that are plant pests? I suppose I was clear.
Yeah, I understood. Your question was if we're using ultrasonic bat waves to kind of, control pests which are of, plants.
Yes.
So this is I know for sure that some places in Costa Rica, some places in Ecuador have actually used, these ultrasound signaling emitters in their fields to ward off pests. But the thing is, a lot of these pests, you can only control the egg laying of females when you use this. But once the eggs have been, eggs are laid, it's very, it really doesn't work because the caterpillars have not, they do not really have a lot of defense, or you cannot deter them from feeding on the plant. So the best thing for, deterring plant, so if you want to deter females from laying eggs, yes, you can actually use these bad echolocation to jam, to scare these females away. But when it comes to larvae, I think the best way or the safest way is to introduce a lot of biocontrol pests like spiders, like ants and parasitoids, which will do the job for you. But there's also a downside of using, bat ecolocators to jam these signals, to scare away moths, because then you're also, polluting the environment with your, echolocation, and that can actually have a lot of effect on native bats. And which will again kind of have a own cascade of its bad effects. So the best, so that's the reason people have not excuse me, have not used bat echolocation a lot. What they've used is pheromones. So they take in the female pheromone or the male pheromone, they apply it to a surface, or they apply it to a container and they hang it in the field. And within a few hours, you have like hundreds and hundreds of moths just sticking onto it, and you can just kill them right away, or you can manage them however you want. And in terms of managing caterpillars, I would definitely say biocontrol pests is much better than using any sort of insecticides, because that is kind of you're kind of leaching the insecticides to the environment. So yeah, that is my opinion on using echolocation for pest control.
Yeah, I think Krenmay would be satisfied. The next question asked by her: if two slug moths come closer, do they die due to their spines? Is the second question she has raised.
So if you talk about two slug moth caterpillars, if they come together, they'll die. No, they won't, because the way the way to look at this is to look at their morphological structures under SEM, so that what kind of structures, physical structures they have, which makes up the spines and what lays underneath it. So the spines have glands which produce these itchy substances. But apart from these openings, everything else is very well covered. So even if you have 10 slug moths next to each other, even if they keep touching it, they keep bashing into each other, they're totally immune to it. It's only us who have, or any other predator which have these specific receptors or specific, combinations of these receptors which will create us to either run away from the predator or to start itching is where it affects. So the moths or caterpillars are totally immune. In fact, if you see a lot of slug moth caterpillars, you can see them huddling in groups. They are gregarious. That means they form groups and they kind of stay together. So it definitely does not affect the moth caterpillars.
Okay. The third question by the same person. If moths are nocturnal, how can they see? Or what about their vision use?
Yeah. So, for example, when I spoke about vision in moths, so you see, people have started to observe, or they've started to discover that from the morphological aspect till the genetic aspect, moths have evolved specific structures to help them see in the night. So, for example, if you look, insect eyes are made out of ommatidia, which are small eyelets, eyes which, when you combine into millions of them, form the insect eye. So if you compare, this is one way which again students can actually do it if they have, once they get proper permission from, forest department authorities or local authorities, where you can collect moths and you can actually spread a thin layer of nail polish, like the clear nail polish on the eyes of the moths, and you can peel that off. And then if you look at it under the microscope, you can actually measure the size of each ommatidia. And what people have seen is that the nocturnal moths have higher or larger ommatidia compared to the diurnal moths. So having larger, structures is one great thing because you can have more light penetrating into it. And secondly, what people have seen is that the cones and rods, which are the unit of eyes which help in perceiving light, they are way more sensitive compared to diurnal moths. So having larger, ommatidia, having more sensitive cones and rods, and your gene expression, the opsin gene expression, is completely different in nocturnal moths compared to diurnal moths. So nocturnal moths have specific opsin gene expression which will express even at the slightest of light. So combining larger eyelet, ommatidia size, very sensitive rods and cones, and differential expression of opsin genes in nocturnal moths, I think they can see very well. And they also have ears to kind of avoid the bats, and eyes to see whatever they have to see. And usually when moths are flying about in the night, they use a lot of navigation systems, such as their surrounding habitat, in terms of what colors the surrounding habitat is, what contrast it is, and what is the position of the sun, and so not the sun, the moon and the stars, so that they can orient themselves. And looking at whenever they see something white, they know, okay, that is something that I can go on and feed or something like that. So moths have very fine-tuned vision equipped for the night, especially nocturnal moths.
Fine. There's a question by Bharati Shanbag. Why moths are available more during the early raining season?
No, that's directly related to host plant availability. Let's say, just before the, it again depends on what kind of, moths you're talking about and what kind of species you're talking about. So there are three ways I don't know if it's three or four, but yeah, there are multiple ways in which this can happen. So let's consider you see a lot of moths right after the monsoon or right during the monsoon. So what this means is that just a few weeks or months before the monsoon, there must have been a lot more foliage, a lot more plants. So there are a lot more eggs or caterpillars survive to huddle to it. So you have a population burst happening. Or you could, that's one condition which can happen right during or right before the monsoon. And then you also have the spring season coming up right before the rainy season, or spring, and you have the spring, the summer, and the rainy. So when you have a lot more younger leaves in spring season, that means younger leaves usually do not have great chemical defense in them. So a lot more, moths can feed on them, a lot more caterpillars can survive. And then when they pupate during summer, they can all emerge in huge numbers during monsoon. And third thing, what you can see is you have these periodical fluctuations where you have a boom of, population burst, and then you have, a down of population happening. That's a cycle which happens in nature, and depending upon species, you can have bursts every 10 years, you can have every burst every few 3 or 4 years. So it again depends on species biology, environmental conditions, and again, plant quality as well. So combining all of those three, you can actually predict when you will see bursts of populations happening in your surrounding area.
Fine. There is another interesting question by
Dr.
Angela
John Paul
Bhattacharya.
will
Yeah.
John.
Dr. John
Yeah.
Paul, Nitin, can we wind by 8? We don't want to take much of your time.
Yeah.
Yeah.
This one
Dr.
interesting
John Paul,
question.
2 more. Yes.
John, 1 second.
Yes, sir.
Nitin, can you put on your video? Let us see you for the last few minutes at least.
Yeah, it's still early in the morning, so let's see if I can make myself. Yeah. I mean, I'm not sure if you can see me now, but yeah. Stop sharing. Yeah.
Yeah, I can see you.
Yeah.
We can see you now.
Yeah.
Fine. See, if moth they pollinate at night, why do they visit our home, especially at night?
Yeah. So think about it this way. So whatever vision that has evolved in moths happened much before the evolution, much before the invention of the light bulb. So all of your light bulbs are bright sources which the moth senses, saying that this is something that I need, that is favorable for me in terms of either laying eggs or either pollinating flowers or getting nectar. So what has happened ever since the invention of light bulb is that in the night it is supposed to be completely dark, but it is not dark. So moths are getting confused that light sources are actually sources of food or sources of mate locations and things like that. So this term, there's actually a term for this, which is known as an evolutionary trap, where signals which were once very favorable, like before the invention of the light bulb, anything that was bright and nice was supposed to be very rewarding for the moth, right? So if you have a very big bright patch of flowers, that means there are more flowers, there's more nectar there, so you can actually eat a lot more. So signals which were once very rewarding are now causing disruptions for those organisms. And this is known as an evolutionary trap because the moths have not evolved any sort of mechanism to avoid these traps because their vision is just tuned to see if it's bright, it's food. So they fall into this trap, and this is a consequence of evolution over a period of time, and that's why we call it an evolutionary trap. So I would, if that's the main reason why moths come into your homes, or they keep flying around fire and they get caught in fire and die.
Fine. There's another question. Will the moths be able to survive below 15 degrees centigrade Celsius, maybe lower temperature?
Yeah. So a lot of the temperate moths that we see, especially North America or Northern Europe or Russia, moths can actually even survive below freezing point. So what happens is that they shut down their system, they shut down their circulation, they shut down their metabolism, and they just lie down, they just stay as still organisms on the snow or wherever they are. And once the temperature starts heating up, they activate their metabolism again and they're good to go. So moths, again, as adults can survive over winter, as by freezing themselves, or they can also do one thing where the adults lay eggs right before the winter, and then when the eggs kind of stay dormant for like 6 or 7 months, and then they hatch when the conditions are all right, or they can feed up till becoming pupae, and then they can become pupae, and the pupae can overwinter, and then once the conditions are all right, they can come in. So moths can easily survive well below freezing points. But again, it all depends on the species of moths you're talking about. If you put a tropical moth, let's say in Canada, I don't think it will survive. But again, if you put a Canadian moth in the tropics, it's not going to survive. So it all depends on how much of environmental variation the moth is used to.
Fine. Now, what is actually the approximate lifespan of the moths? The next question.
That depends entirely on the species you're talking about. Some of the moths could just be alive as adults for a day. They can be alive as adults for a month or 2 months or 6 months. Some can be they can live as larvae for like 1 year or 2 years. Some can live as larvae for 3 years. Some can live as pupae for 3 years. So it again depends on the kind of species you're talking about and the kind of life stage you're talking about. but you can have a huge range of variation from just a few days to almost a year or something.
Fine. Now there is another interesting question. We spoke about the toxic moths.
Yeah.
Yeah. Now, how do they attain the toxicity? Is it by only their feeding mechanism, or by what other means? Can you give a few other examples?
Okay. Yeah. So the evolution of aposematism is again a very interesting concept, both in ecology and zoology as well as chemical ecology, because you can tackle this issue from a lot of different angles. So if you let's talk about I'll try to say this in the form of a story. So imagine a species of moth has started feeding on a plant, and the plant starts evolving defenses, and the moth again does something, evolves something else to counter defense. So after a period of time, you have this plant which is very, very specialized, which is producing this very nasty chemical which only this moth can feed. So there are two things that the moth can do. So one is it can find a way to detoxify this chemical compound in some through its metabolism, where let's say if you eat cyanide, you just attach a side chain to it and then make it it's no longer toxic. Or you can just change the chemical structure of it and then it's gone, it's no longer toxic, and then you can happily feed on it. And second, what you can do is you can take that compound and store it in organs and encapsulate it in such a way that it's still very toxic, but it is stored in very specific organs in your body. But let's say if that thing goes away from that particular organ, you will also die. But as long as it's there in that organ, you're safe, and it is toxic. And since you store that toxic, anything that tries to eat you will will eat that toxin as well. So that is how moths kind of store those chemicals, where they have specific structures to store them. So I mean, last year I was involved in a study where we published this research in grasshoppers, where we found out that there is this channel called a sodium potassium channel. This is very, very important in all organisms throughout life to maintain your electrolyte balance, to maintain your electrical conduction. So what happens in these grasshoppers is that they push out everything outside the sodium potassium pump channel, and they protect the sodium potassium pump, because these toxic compounds attack the sodium potassium pump and they just destroy the sodium potassium pump. So by protecting the sodium potassium pump, these grasshoppers are able to feed on these toxic plants and themselves become toxic. The same thing with moths. So they might have specific adaptations to sodium potassium pump, and if they are feeding on cardiac glycosides, if they're feeding on iridoid glycosides, they might have some other sort of protection. So it all depends on what kind of chemical compound you're talking about, where that chemical compound has a effect on the body, and how do you protect that particular thing. And once you have evolved these characters, you can easily feed on the toxic compounds, either detoxify them and leave them, or store them for yourselves and become aposematic, and then make sure that no one else eats you as well.
Fine.
Does that answer the question?
What is the reason? Yeah. What is the reason for the moths to get attracted more towards light other than the other insects? In general.
Yeah, that I mean, like I said, it's like when you have very high sensitivity for if you have evolved in during if you have evolved in nocturnal vision, that means you're very sensitive to whatever small changes of light is. So things which have evolved in in bright light in the morning, they know that they have light all around, so they can kind of discriminate what is better and what is bad. Whereas in the night, you do not expect to see any light, and all of a sudden you have light. That means you kind of go into it. That's one of the reasons why moths kind of get attracted more towards light than any other organism, because they have not experienced any other form of light, just because they are only seen during the night. And you need to think about it in terms of what happened, what the world was like before light bulbs came in, what the world was before humans started using fire for light or anything like that, because moths have been there for 300 million years. I mean, that's a very, very long time. And all of these basic mechanisms have evolved so far back in time that they are not going to change just because humans have been here for 1,000 or 10,000 years. I mean, it might take another few million years to actually change those mechanisms, and I'm not sure if humans are going to stay that longer anyway. So, yeah.
Oh, fine. Now, the moths, they lay eggs, no?
Yeah.
They lay eggs, either in the wall or on a cloth or on the plant sources. Is there any other way out for them to lay eggs?
Dr. John Paul.
Then we
Sorry
will
to
ask.
interrupt.
Yeah.
What we'll do is, if there are questions, let them post it to us, and then we'll send it to Nitin, and Nitin can answer them over mail because it's getting late. We will leave it, pass on to Dr. Kavya for the vote of thanks. Thank you, Dr. John Paul, for moderating.
Yeah, and
Dr.
before, and before we end, I mean, again, especially for students, if you want to contact me, I mean, I am here because I got a lot of help from all the faculty that that taught me in Joseph's, and after that as well. So I'm more. I mean, I know you have a lot of support from your college, and I'm more than happy to help you guys plan studies, because I know your professors definitely help you go ahead with it. And if any students are interested in contacting me about learning more about science or more about anything, or I'm more than happy to do it. But just to say that my specialization is in ecology and evolution, so if you ask me more about developmental biology, I might not know more than what Shiv Kumar sir taught me. So yeah, that is where my knowledge would be. But if you wanted to learn more about ecology and evolution, I'm more than willing to talk to you guys about it.
Dr. Kavya is there?
We are not able to hear you.
Now audible?
Kavya, unmute.
Unmute yourself.
Hi, Nitin. Wonderful talk.
Thank you. Thank you, sir. Thank you so much.
Maintained your standard.
Yeah, your product, sir.
Okay. I think it's getting late. I will pitch in. It's moment to thank Nitin. Nitin, thank you for sparing your time. Sorry for all the glitches that happened. That you were here to present the talk on moth ecology and evolution. All the questions and other queries, we'll pass it on to you. And if students are willing to get in touch, we will try to reach and connect you to the students. So thank you very much for being the resource person for this international webinar on moth diversity, ecology, and conservation.
Yeah,
This
thank you
is
so
also
much.
time. Thank you. This is also time to thank the management, St. Joseph's College, Reverend Father Rector, and also Father Principal for their continuous encouragement and support for us to organize these webinars that are benefiting students. And this coincides with the National Moth Week, and aptly we decided to have this. Thanks that we got two lucky resource persons. We were lucky to get them to have their views presented here. I am also thankful to the Department of Zoology, my HOD, and other colleagues in the department for their support in successfully conducting this webinar. This would not have been possible if so many participants had registered. I understand due to the technical glitches, some of them had to drop. But even then, whoever continued to stay with us, thank you for your participation. Please do participate in webinars that we are planning to host in the coming weeks as well. Last but not the least, this certainly would not have happened without the volunteers of the National Science Association. The pandemic has provided a situation where the previous year, the 1920 batch of National Science Office bearers are still here, and there is this next batch of volunteers also. So it's being a coordinated effort of every activity that they have done. I should thank Rachel Soumya especially because she pitched in to shift this platform from Google Meet to Microsoft Teams. So I'm thankful to every volunteer. The list is 20 plus. I
We
hope
aren't able to hear you.
I think the connectivity touched me
So,
also. Fine. So that's about the vote of thanks. Yeah. Dr. Kavya wants to pitch in. We are not able to get her.
yeah.
Okay. So that's it. Thank you all for participating. Good night. So we'll meet in some other webinar that we have.
Thank you.
Thank you.
Thank you all.
Thank you all.
Thank you to all.